Hello, my name is Shri, and I am a senior in the bioengineering program here at the University of Colorado, Denver. And I'm currently working as an undergraduate researcher at the MYKC lab in the Linda Cernick Institute for Down Syndrome Research here on the Anschutz Medical Campus. So today I'm going to take you through the process of PCR, which stands for polymerase chain reaction. And PCR is a technique used to make numerous copies of a specific part or segment of DNA. And it can target the specific sequence of DNA that you are interested in and make multiple copies of that specific segment so you can conduct further testing on that specific segment. Because DNA is really small and it's really long. And so we need to make a lot of it to be able to visualize it with the naked human eye. And we need to be able to take the part of it that we want to see because it's so big. So, for example, genotyping is the process I'm going to use PCR for today, and with genotyping, we're going to identify different mouse models used for Down syndrome research. So I'm going to figure out which mice are wild type, which in summary means these mice have the same characteristics as we would observe in the wild, and then which mice have Down syndrome. And by identifying the genotype of the mice, researchers trying to learn more about Down syndrome can observe their characteristics and advance their research, which will help persons with Down syndrome in the future. So, to start this, we first need to get DNA. Let me take you through the process. So, to get mouse DNA, we need to go to the source, and the source is the tissue from the animal. Today, I'm going to use very small clippings of mouse tails and ears. The microcentrifuge tube on the left has a tail sample, as you can see in the bottom, and the microcentrifuge tube on the right holds an ear sample. So before I do anything else, I set the heating block to heat to 95 degrees Celsius. And it usually takes a few minutes, so I usually do this first to save some time. As the heating block is heating up, to extract the DNA from the tissue, we need to add extraction buffer to the sample first. So 75 microliters of extraction buffer for tail samples, as I'm adding here. And then for ear samples, we need to add 50 microliters of extraction buffer. And we need to heat up the samples to 95 degrees C so that the DNA can come out of the nucleus and the cell membrane and be in the solution. Once there's extraction buffer in all the samples, I am going to add these caps to the microcentrifuge tubes, as you can see here. And these caps are going to help the microcentrifuges from popping open when I put them on the heating block. So once the heating block has reached 95 degrees Celsius, as you can see right there on the thermometer, I am going to start adding the samples to the heating block. And once the samples are on the heating block, I'm going to start a 30-minute timer. So while the samples are on the heating block, I'm going to prepare the PCR mixes. So one PCR mix will help conclude that the samples are indeed from a mouse, and the other will help conclude whether the samples show a Down syndrome model mouse, which I will start referring to as DP16 from now on, or if it's just a wild-type mouse. Here you can see both of the PCR mixes in their respective microcentrifuge tubes. So the OI-MR mix is going to identify whether the sample is from a mouse or not, and the DP16 mix is going to identify whether the samples are DP16 positive or not. So if it's DP16 positive, the mouse is a Down syndrome model mouse. The PCR mixes are going to contain a few different elements. First, primers, which will attach to the specific part of the DNA that we want to amplify or multiply, because it has the identifying information that we want at that spot. Second, Taq polymerase, which is a DNA polymerase that will make more copies of the DNA once the primers attach. And finally, just free-floating nucleotides. And nucleotides are the building blocks of DNA. And since we want to make multiple copies of DNA, we need these nucleotides in the solution so DNA polymerase can build these multiple copies. Once the samples have been sitting at 95 degrees C on the heat block for 30 minutes, I'm going to take them off and remove their caps. And after the caps have been removed, I need to add stabilization buffer to the samples, because the extraction buffer is there to bring out the DNA from the nucleus and the cell membrane. So if it stays in there for too long, it might degrade the DNA as well. So 75 microliters of stabilization buffer for the tail samples and 50 microliters of stabilization buffer for the ear samples. After the stabilization buffer was added, the sample digest was ready to use. So I added the PCR mixes to each of the samples and placed them into PCR tubes that I just placed into the thermocycler. So what the thermocycler is going to do is cycle through multiple temperatures, and cycling through these temperatures is going to help all the elements in the PCR mix interact with the sample. And after the product comes out of the thermocycler, we are going to have tons and tons of DNA that we want to look at. The thermocycler runs for about an hour, and while it's making multiple copies of DNA, I'm going to prepare an agarose gel. And with the gel, we're going to be able to visualize the PCR product, which is just multiple copies of the DNA of interest. So the OIMR mix PCR product should have multiple copies of a section of DNA that will tell us that we have mouse DNA. And the DP16 mix PCR product should have multiple copies of a section of DNA that will tell us if we have DP16 mice or wild-type mice. To start off to make the gel, I'm going to take 2 grams of, or measure out 2 grams of agarose in this 500 milligram beaker and add 200 milligrams of TBE buffer. And after I add the buffer, I'm going to add the beaker into the microwave for about 2 minutes. And when there's around 30 seconds left, I usually take the beaker out to stir and make sure that everything is evenly distributed. Let it cool down for a little bit and then take it out. And if you look inside, it's not fully clear, and that's when you know it's not ready to be poured yet. So going to put it back in the microwave for probably a minute, and after it's in there, you can see that it is completely clear. I'm going to just touch and make sure it's not too hot to take out, wait for it to cool, but look at that clear gel or liquid that I will pour into a gel. So now I'm just waiting. I waited for the liquid to cool down for about 5 minutes, and then I'm going to add Cyber, which is a form of dye that's going to allow for us to see the DNA when we go to image it later. And after that's added into the liquid, I'm going to pour it into the gel mold right there and wait for that to cool down. And make sure there are no bubbles because that will mess up with the imaging later on. And I usually put the gel in and around 36 minutes before the PCR cycle is ended, so I have enough time to prepare the samples and for the gel to actually settle and harden. So PCR has ended, and I just took the samples out of the thermocycler, and now I am adding 10 microliters of the sample into each one of these microcentrifuge tubes. Just fast forward pipetting for you here. After adding all the samples into each of the microcentrifuge tubes, I'm now going to add DNA dye. So this dye is going to specifically bind to the DNA, and when we add it to the gel, we'll be able to see it. So here, I'm just going to fill up the gel box with TBE buffer and remove those combs. After I remove the combs, you can see the wells lined up against the red tape under the PCR box right there. And now I'm going to take the dye and PCR sample from all of the microcentrifuge tubes and load it onto the gel. So as you can see, I'm adding it into the well. This is actually one of the harder techniques because you have to make sure your hand is really steady, but thankfully after doing it for multiple times, it becomes one of the easier things to do. So now we are going to see a fully loaded gel right there. You can see all the samples in their little wells right there. And now I'm going to complete the setup of the gel box by adding this cap, which has 2 electrodes at either side, the black being the negative side and the red being the positive side. And since DNA is negatively charged, it's going to move to the positive side. And this box is going to help pass a current through that will allow the DNA to move towards the right side. And how do we know that the current is there? We see these bubbles on the sides, and this ensures that the gel box is on and ready to go. So after the gel finished running, I was able to take it to the imager, and these are the results. So the top row is going to show results from OIMR, and the bottom row is going to show results for DP16. So in the top row, as you can see, the first lane, marked with a negative, is the negative control, which means that we should see nothing there. And if we see something there, that means something went wrong with the experiment. And then there's the ladder. So DNA is measured in base pairs, and the ladder is what's going to help us determine what size our DNA is. And so we have the ladder as the second lane, and the third lane is the positive control. And the positive control, it better show up, or you again did something wrong in the experiment. And the positive control is where your samples should be. So we're going to compare all our samples to the positive control. And the third lane is positive control. So for OIMR, where the primers binded and cut the DNA and the DNA was replicated, the size should be around 350 base pairs. So as you can see, compared to the ladder on the side and the ladder on the gel, the DNA for OIMR shows up at just about 350 base pairs. And remember, OIMR was just showing if this sample is from a mouse or not. So since all of the little bands show up for all of the samples, we know that yes, this sample is from a mouse. Then we move down to the second row, which is the DP16 row. And for DP16, the samples should show up around 355 base pairs. And as you can see, compared to the positive control, 1, 2, 3, 4, 5 of the samples show up. So sample 1 and sample 3, 4, 5, and also sample 9 are DP16 positive, which means all those samples, the positive samples, were obtained from mice that have Down syndrome. In the DP16 section, the lanes without any bands show mice that are wild type. So DP16 positive is with bands and DP16 negative is without the bands. And now we know which mice are Down syndrome models and wild type. That is the result of genotyping. Thank you guys for sticking around and watching this video about PCR and genotyping. Hopefully this was really helpful and it made sense, and I hope you have a good rest of your day.